So today we will discuss our new topic, which is basically the role of intron and exon in gene expression. Plus we'll be doing this book reading as well, because usually the MCQs arrives from the books and we need to read them as well to make our concepts more clear, as these topics are new and are not found in the previous book, as this year the book has been changed and we need to continue with the new curriculum as well. So let's start with this. So first, the role of intron and exon in the gene expression. So basically, as we know that in basically in prokaryotic organisms, in prokaryotic organisms, introns and exons are not present. Introns are not present. Why? Because there is basically absence of split genes. That's why the introns and exons are not present in prokaryotic. But in eukaryotic, these introns and exons are present. Introns and exons both are present in the eukaryotic living organisms, right? So now here let's discuss this. Here this is written over here that the eukaryotic organisms, basically the eukaryotic genomes contain introns and exons, while prokaryotic gene does not contain intron. During gene expression, all introns are transcribed into RNA and replicated during replication process, but introns do not participate. Okay, here the important thing is written that the introns are transcribed into RNA. Here an important thing is written that these introns are transcribed, plus they are replicated as well, but they do not participate in translation. They do not do translation. These introns do not do translation, but they do replication, transcription, because before coming out of a nucleus, the introns regions are eliminated by spliceosome. Okay, now what is this? Basically, as we know that when there is a process of these introns plus these exons go through the process of replication plus transcription, but in the result of transcription we get pre-mRNA, which cannot move from the nucleus. So in order to convert this into the exonic RNA, the process of basically post-transcriptional, you can say the splicing, tailing and capping takes place. So that's why this pre-mRNA is converted into exonic RNA and introns are eliminated. So these introns cannot do the process of translation, right, are eliminated by the spliceosome. So as we know that do you think that these introns are basically junk? They are basically not junk. The 40% of the intron are 40% of the intron, and on average 40% of intron is present in our genome. Now this question arises here, why introns are present in genome, right? So the the existence of introns in genome is a real mystery, given the expensive energy cost for a cell to pay for copying the entire length of several introns in a gene and eliminate them at the exact position introns. Okay, now they are saying that when in the last in transcription they are eliminated, so why so much amount of energy is utilized by the process of replication and transcription? Like there's a lot of 40% of our genome is introns, so why this 40% is basically is being replicated, it's been transcribed, why they waste so much kind of energy? Because so is because this introns are totally junk, right? According to them, this is a question that introns are totally junk, so why should we use this 40% of why you we should use a lot of energy in order to basically convert to in order to basically replicate and transcribe these introns, which are ultimately which are being eliminated at the end, which are not going to be translate. So why? So the answer is, so it has come to know that introns are not totally junk. So basically these introns it is these introns are not totally junk. These are crucial because protein variety is greatly enhanced by alternative splicing, in which introns play important role. The alternate splicing is controlled molecular mechanism for synthesizing multiple variant proteins from a single gene. Okay, what does that mean? Basically there is a process of, okay, basically these introns play a very important role in alternate splicing. Alternate splicing, what does that mean? Okay, consider here we have this is 5 prime to 3 prime. These are exon 1, this is intron 1, this is exon 2, this is intron 2, this is exon 3, and this is intron 3. Okay, we know that in the process of basically splicing, what happens, the process, they basically synapse protein come and spliceosome is formed and introns are basically eliminated. So we say that in alternate splicing, this is 5 prime to 3 prime. First of all, what will happen? First exon 1 will come, then exon 3 will come, then exon 2 will come, right? Then exon 2 will come. Basically this is not in sequence. It means that intron 2 has taken some time to get spliced, right? So because of this splice we got different protein. Consider we got the topomin protein alpha. Topomin alpha by this, but by 5 prime to 3 prime, exon 1, exon 2 and exon 3. So we have gotten topomin beta. So by this alternate splicing, like by the replacing first exon 2, then after 3, so we are getting different kind of proteins. We are getting the different proteins. So this is basically this is basically important role which introns are playing. If introns will not be the part of this DNA, then how we will be getting the different proteins? So by this alternate splicing, basically we are getting this different kind of proteins. Now we have another. Okay, introns play important role in the positive regulation of gene expression. It was found that without introns, the protein products were significantly diminished. It was also found that some introns are designed to construct expression vectors for guaranteed a high level expression compared to gene without introns. Okay, basically you can say a competition was held in which two DNAs were being observed, in which one there were introns, and this DNA does not contain the introns. After we said that after the protein is being formed, it was observed that the DNA which was having the introns produced more protein than the DNA which was not having the introns. So basically this is basically the significance of introns. Okay, you can also say that basically in introns, the first intron, this is especially first intron. The first intron is always a long intron. This long intron basically gives signal to RNA polymerase to join transcriptional initiation factors. It basically, this long intron gives signal to the RNA polymerase to join the transcriptional initiation factors, right? So I hope this is also clear. With introns, the introns are involved in transcription, initiation and termination process. These processes require some sequence elements in intron in correct order. Introns may be associated with mRNA transport or chromatin assembly. Introns also play some indirect role. The first introns play important role in transcription. As I told you, it has been found that the first intron is the longest intron, which works as a signal for the transcription. So many other roles of introns are going to be established day by day in research in molecular biology, right? So this was basically this was all about the role of intron, exon in gene expression. So I hope this is clear to you. If you have any kind of queries, you can ask me in the comment section. I would be really glad to answer you guys.